If you haven't heard about the dangers of microplastics, then you really are living under a rock. And so in today's video, what I want to do is share with you the dangers of microplastics and first of all, what we can actually do about it. Before we get into the video, let's understand a little bit about microplastics. Now, these microplastics are small plastic particles and they're less than 5 millimeters in size that come from the breakdown of larger plastics or are intentionally manufactured like microbeads. Now, these particles are widespread in the environment and can be ingested by both wildlife and also humans. And the microplastics consist of various types of plastic and can also contain chemicals such as BPA and forever chemicals such as PFAs. Now, a common misconception is that BPA is the same as microplastics. However, BPA is a chemical found in certain plastics, and while it can be a part of a microplastic, it can also leach out of plastics before they even break down into microplastic form. Now, BPA can mitigate and migrate from plastics into the environment during use, posing health risks long before plastics degrade into smaller particles. In this video, we'll look at potential mechanisms by which specific types of fiber that we consume may help to actually remove BPA and microplastics from the body. So here we can see the dangers of microplastics. We can see that it can affect the brain, it can affect muscles, skeletal muscles, platelet aggregation, tissue accumulation in the lungs, it can affect testosterone levels, it can affect fertility, it can accumulate in the kidneys, it can deplete microminerals, it can also deplete the important pathways and cause dysfunctions in the GIT, gastrointestinal tract. Clearly, microplastics are damaging to overall human health. Now, how long do they actually last in the human body? We actually don't know yet how long microplastics stay in the body, and the exact duration can depend upon factors such as type of plastic, the size of the particles, and individual differences in metabolism and excretion. And research suggests that microplastics can persist in the body for weeks to years and are generally micro and nanoparticles that can be excreted from the body through various pathways, including the bile, the urine, the feces, and in some cases, sweat, saliva, tears, and breast milk. Now, while their size plays a crucial role in determining the excretion route, larger particles tend to be expelled via the digestive system. Smaller ones can pass through the kidneys, but further research is needed to fully understand these processes and their health implications. So let's hear what Dr. Rhonda Patrick has to say about microplastics.
How can I get rid of them? I got too many microplastics in my body. How do I get rid of them?
Yeah, so microplastics, we kind of talked about a little bit getting rid of them, and that really the main source is excretion through feces, and that happens with dietary fiber. So if you are eating a lot of fruits and vegetables, that does increase the chances that microplastics are going to move their way out through feces
Is
more
that
readily.
only microplastics that you're consuming along with it, or is it able to pull microplastics, sort of detoxify them from your body?
I think it's in general, to be honest, because if you think about like fiber, what does it do? It moves stuff through your body. It doesn't necessarily have to be something that you just ate, right? Like it could be something that you had hours before. So if you're getting like daily fiber, right? Like thinking about getting your total daily fiber, I think that's something that is important because it's moving it out of your body.
So clearly we can see that Rhonda Patrick is actually suggesting to increase fiber intake through fruits and vegetables, which helps to detoxify the body of these microplastics.
So you can absorb it through both ways, like the ways of just moving it through, but also with the type of fiber that's found in things like, berries and apples. So these are pectins, like inulin, all this type of fermentable fiber, green bananas, resistant starch, like that stuff makes viscous gel-like substance in your gut, which encapsulates microplastics so that you're not absorbing them. So really, I think it's just like more important to focus on the daily fiber intake versus with a meal.
So is that the biggest mover, daily fiber intake, do you think?
I do, and this is all animal data. Like this is all new. There's no real human data yet.
We can see that clearly there's only a small amount of recent studies on this particular subject, but let's analyze what she says around fiber ingestion and microplastics. So looking at the absorption and sequestration in the gut. So looking at how microplastics can be adsorbed. Sorry, let me just correct that. That's adsorption, which is the opposite of absorption. It's adsorption and sequestration in the gut. Fiber can physically bind or adsorb microplastic particles in the digestive tract, preventing them from contacting the gut lining or being absorbed. Dietary fibers have a porous structure and large surface area that enables them to trap exogenous particles. Chemical interactions play a role too. Fiber surfaces carry functional groups that can form electrostatic attractions and hydrogen bonds with plastics. For instance, positively charged amine groups on microplastics such as certain nylon or polystyrene particles will stick to negatively charged sites on fiber. Hydrophobic interactions also help fibers, especially those with aromatic components, bind plastic fragments. By acting like a sponge or adsorbent, fiber in the intestinal contents can latch onto free-floating microplastics. The entrapped microplastics then get carried out of the body when the fiber is excreted in feces rather than lingering in the gut. This mechanism was observed in vitro through a study we'll talk about where coffee grounds pulled microplastics out of a solution, and is strongly presumed in vivo. Essentially, ample fiber provides more binding sites to capture microplastics, reducing the fraction that remain in the gut or pass into the bloodstream. In addition, we can see that increasing fiber intake can increase stool bulk and cause a faster transit. Insoluble fiber increases stool bulk and speeds up intestinal transit, reducing microplastic retention in the gut by diluting ingested particles, minimizing their contact time with tissues, and promoting regular excretion through a mechanical flushing effect. There's also enhanced mucus barrier and gut integrity. Soluble fiber enhances the intestinal mucus barrier and gut integrity by promoting short-chain fatty acid production, strengthening cell junctions, and reducing permeability, which helps to trap these microplastics, preventing their absorption and facilitate their excretion. There are other potential mechanisms such as degradation by specific gut microbiota. However, there is lack of research in this particular area. If you're someone that's been trialing different supplements and you're still lacking that energy, spark, drive, and overall vitality, then you may want to check out my brand new supplement that I've just released called Catwa Pure. Catwa Pure harnesses the power of a particular Amazonian herb known as Catwaba bark, which has been used for centuries to boost mood, enhance energy levels, and act as an aphrodisiac. So definitely check out Catwa Pure. You can learn more by visiting inbeforesups.com. Fiber ingestion and microplastics, the research. Here's the first study. Studies analyzing dietary fiber intake and microplastic excretion in humans is very limited. And as a result, most of the evidence is pieced together from animal studies. Let's take a look at the research. A study investigated whether dietary chitosan supplementation could enhance the fecal excretion of dioxins and polychlorinated biphenyls in 6 healthy men by administering either 0, 1, or 3 grams of chitosan daily over a 4-week period while monitoring fecal toxin excretion. Chitosan derived from chitin in shellfish exoskeletons functions similarly to dietary fiber in that it is indigestible and increases fecal bulk, but unlike plant-based fibers, it carries a positive charge that enhances its ability to bind to fats and fat-soluble toxins. The results showed that consuming 3 grams of chitosan significantly increased the fecal excretion of polychlorinated biphenyls and dioxins, with a strong correlation between toxin excretion and fecal fat content, suggesting that chitosan binds to lipophilic pollutants and promotes their elimination. The next study on this particular subject is looking at another study on mice examined whether dietary psyllium seed gum could reduce bisphenol A absorption and its estrogenic effects in female rats by dividing them into 3 groups. One group receiving a standard diet, one consuming a diet with bisphenol A, and another consuming bisphenol A with 5% psyllium seed gum for 6 days. The results showed that rats in the bisphenol A group had significantly higher uterine weights than the control group, but those receiving psyllium seed gum had significantly lower uterine weights and bisphenol A levels in the serum, liver, and uterus compared to the bisphenol A only group. Fecal bisphenol A excretion was significantly higher in the psyllium seed gum group, indicating reduced intestinal absorption. These findings suggest that psyllium seed gum can mitigate the estrogenic effects of bisphenol A by preventing its absorption and promoting its elimination through feces. This next study investigated whether a buckwheat-enriched diet could mitigate the oxidative stress caused by BPA in male Wistar rats by dividing them into 4 different groups. One group receiving a standard diet, one consuming a diet with bisphenol A, one receiving a diet with 30% buckwheat, and one receiving both bisphenol A and a buckwheat-enriched diet. The results showed that the rats consuming bisphenol A exhibited increased oxidative stress markers, but those on the buckwheat diet had significantly improved antioxidant enzyme activity, reduced oxidative damage, and enhanced levels of sirtuin-1, a protein associated with stress resistance and longevity. Importantly, sirtuin-1 was not impacted by BPA exposure at the level given to the rats. However, buckwheat enhanced sirtuin-1 in groups ingesting it. Although fiber excretion was not specifically measured, buckwheat contains approximately 10% fermentable dietary fiber, which may contribute to its protective effects. These findings suggest that dietary buckwheat can counteract bisphenol A-induced oxidative stress by modulating antioxidant status and sirtuin-1 levels, highlighting its potential as a protective dietary intervention. And another study done on mice investigated the impact of microplastic exposure on hypertension and cardiovascular health, comparing groups exposed to microplastics alone, microplastics plus a high fiber diet, and microplastics plus antibiotics. Mice exposed to microplastics showed increased blood pressure, gut microbiota alterations, and cardiovascular damage, including myocardial hypertrophy and fibrosis. But these effects were significantly reduced in the group receiving a high fiber diet, which promoted beneficial gut bacteria and enhanced intestinal barrier function. Histological analysis revealed that fiber intake mitigated vascular thickening, cardiomyocyte hypertrophy, and fibrosis, while also increasing goblet cell density in the intestinal lining, suggesting improved gut health. These findings indicate that dietary fiber can counteract MP-induced hypertension and cardiovascular damage, with authors suggesting the mechanism was through modulating the gut microbiome, reducing systemic inflammation, and enhancing intestinal defense mechanisms. While this is certainly a plausible mechanism, potentially fiber consumption also enhanced the secretion of microplastics. And then finally, a study investigated whether coffee grounds could remove nanoplastics from water by testing their ability to adsorb fluorescent polyestrine nanoparticles under varying conditions, including pH, coffee ground concentration, initial nanoplastic concentration, and contact time. The results showed that coffee grounds effectively captured nanoplastics across a wide pH range, with a maximum removal efficiency of 74%, achieved at a concentration of 25 grams per litre and a contact time of 40 minutes. Adsorption followed the Dubinin-Radushkevich isotherm and pseudo-second-order kinetic models, indicating that nanoplastic removal was driven by electrostatic interactions and hydrogen bonding between the surface functional groups of coffee grounds and the nanoplastic particles. Given that dietary fiber, particularly insoluble fiber like that found in coffee grounds, has been shown to bind various toxins and promote their excretion, these findings suggest that fiber consumption in humans could play a role in trapping and eliminating microplastics and nanoplastics in the digestive system before they are absorbed into the bloodstream, potentially reducing their long-term health risks. So here are my final thoughts. I think that fiber ingestion could be a viable strategy to help mitigate the harmful effects of BPA and xenoestrogens. We do need more research in this particular area, and whilst this research is exciting, it does show promise for a potentially balanced diet, and actually those that eat only a meat-based diet could be at risk of accumulating more microplastics. And in addition to this, things like probiotics, sauna usage, and sweating, and fiber now are showing promise at helping to mitigate and shield the body against these harmful toxins. That's it from me today, guys. I look forward to seeing you in the next video.